Hello everyone, my name is JOYCEREL from BS Ed Science, and today I'm going to discuss Chapter 11, Food and Industrial Microbiology, Microbiology of Food and Applied in Industrial Microbiology. First, let's start with the key terms. Fermented foods. Foods that have been intentionally altered during production by carefully controlling the activity of bacteria, yeast, or molds. Food preservation. Increasing the shelf life of foods by preventing the growth and concurrent metabolic activities of microorganisms. Food spoilage. Biochemical changes in foods that are perceived as undesirable. Food infection. An illness that results from consuming microbes in a food product. Ingested microbes colonize the host and cause disease. Foodborne intoxication. An illness that results from the consumption of exotoxin produced by microorganisms growing from food product. Water activity. The relative amount of water available. Pure water has water activity of 1.0. So we are going to discuss the factors influencing the growth of microorganisms in foods. 2. Microorganisms in foods and beverage production. 3. Food spoilage. 4. Foodborne illness. And last but not the least, 5. Food preservation. So first, let's discuss the factors influencing the growth of microorganisms in food. So the first factor is intrinsic factors. So under intrinsic factors is the water availability. Food products vary dramatically in terms of how much water is accessible to microorganisms, fresh meats, and milk. For example, have ample water and will support growth of many microbes. Bread, nuts, and dried foods, on the other hand, provide a relatively arid environment. Some sugar-rich foods such as jams and jellies are seemingly moist, but most of that water is chemically interacting with sugar, making it unavailable for use by microbes. Have salted foods, for similar reasons, have little amount available moisture. The term water activity is used to designate the amount of water available in foods. By definition, pure water has water activity of 1.0. Most fresh foods have water activity above 0.98, whereas ham has an water activity of 0.91. Jam has water activity of 0.85, and some cakes have a water activity of 0.70. Most bacteria require a water activity above 0.90 for growth, which explains why fresh, moist foods spoil more quickly than dried, sugary, or salted foods. Fungi can grow at water activity as low as 0.80. which is why forgotten bread, cheese, jam, and dried foods often become moldy. Staphylococcus species, which are adapted to grow on one dry, salty surface of human skin, can grow at a water activity of 0.86, lower than the minimum required by most common spoilage bacteria. Staphylococcus species normally do not compete well with other bacteria, but on salty products, such as ham and other cured meats, they multiply with little competition. Ham, when improperly handled, is a common vehicle of Staphylococcus aureus food poisoning. So the second intrinsic factors is pH. pH of the food is also important in determining which organism can survive and thrive on it. Many species of bacteria, including most pathogens, are inhibited by acidic condition and cannot grow at a pH below 4.5. An exemption in it is the lactic acid bacteria, which can grow at pH as low as 3.5 and are used in the production of fermented foods such as yogurt, sauerkraut. This group of bacteria produces lactic acid as a result of fermentative metabolism. Although they are useful in food production, their growth and accompanying acid production is also a prime cause of spoilage of unpasteurized milk and other foods. Fungi can grow at a lower pH than most spoilage bacteria, leading to some acidic foods eventually becoming moldy. For example, the pH of lemon is approximately 2.2, which inhibits the growth of bacteria, including the lactic acid group. But some fungi can grow. The pH of food product can also determine whether toxins can be produced. For instance, Clostridium botulinum, the causative agent in botulism, does not grow or produce toxin below pH 4.5, and so it is not considered a danger in highly acidic foods. This is why the canning process for acidic food and pickles is less stringent than that for foods with a higher pH. However, some newer varieties of tomatoes are less acidic than older types and require the addition of acid if they are to be safely canned using these less stringent procedures. Third intrinsic factors is nutrients. Nutrients present in the food determine the kinds of organisms that can grow in it. An organism requiring a particular vitamin cannot grow in a food lacking that vitamin. A microbe capable of synthesizing that vitamin, however, can grow if other conditions are favorable. Members of the genus Pseudomonas often spoil foods because they can synthesize essential nutrients and can multiply in various environments, including refrigeration. So the fourth is biological barriers. Biological barriers, rinds, shells, and other coverings help protect foods from invasion by microorganisms. Eggs, for example, retain their quality much longer with intact shells. Whole lemons keep longer than slices. Even so, microorganisms will eventually break down these coverings and cause spoilage. And the fifth factor is antimicrobial chemicals. Antimicrobial chemicals, some foods contain natural antimicrobial chemicals that help prevent spoilage. Egg whites, for instance, is rich in lysozyme. If lysozyme susceptible bacteria breach the protective shell of an egg, they are destroyed by lysozyme because they can cause spoilage. Other examples of naturally occurring antimicrobial chemicals are benzoic acid, which is found in cranberries, allicin in garlic, and an antibacterial peroxide system similar to that found in phagocytes, which is in raw milk. So the second factor that influences the growth of microorganisms in foods is extrinsic factors. So intrinsic factors is the extent of microbial growth varies greatly depending on the condition under which a food is stored. Microorganisms multiply rapidly in warm, oxygen-rich environment just as the surface of meat stored at room temperature. So storage temperature is the temperature of storage affects the rate of growth of microorganisms in food. Below its freezing point, with water crystalline and inaccessible, effectively halting microbial growth at low temperatures above freezing. Many enzymatic reactions are either very low or non-existent, with the result that some microorganisms are unable to grow. Those that can do so at a reduced rate. Microorganisms that grow on refrigerated foods are most likely psychrophiles or psychotrophs, such as some members of the genus Pseudomonas. Second, under intrinsic factors is the atmosphere. The atmosphere is the presence or absence of oxygen affects the type of microbial population able to grow in food. For example, members of the genus Pseudomonas are obligate aerobes, and consequently, they cannot grow in food stored under conditions that exclude all of their required oxygen. Excluding oxygen from a food, however, may enable the growth of the other bacteria, including obligate and aerobe Clostridium botulinum. A case of botulinism was traced to consumption of a thick homemade stew that had been slowly cooked and then left at a room temperature overnight. The cooking process did not destroy the endospores of C. botulinum and had created an aerobic condition in which the organisms thrive and produce toxins. So let's move on to microorganisms in food and beverage production. So, under the microorganisms in food and beverage production is the lactic acid fermentation by the lactic acid bacteria. So first is cheese, yogurt, and other fermented milk products. The tart taste of yogurt, pickles, and sharp cheese, and some sausages is due to the production of lactic acid by species of Lactobacillus, Lactococcus, Streptococcus, Leuconostoc, and or Pediococcus. The starter cultures and sometimes rennet are added to pasteurized milk to make cheese. Other bacteria or fungi are sometimes added to ripen cheese to impart characteristic flavors or textures. Another is pickled vegetables. Fermentation of vegetables pickling relies naturally occurring lactic acid bacteria. Third is fermented milk products. Commercial sausage production utilize starter cultures to rapidly decrease the pH and prevent the growth of pathogens. These are the lactic acid fermentation that uses lactic acid bacteria. Next is alcoholic fermentation by yeast. First is wine. Wine is the product of alcoholic fermentation of naturally occurring sugars in the juices of fruit by specially selected strains of Saccharomyces cerevisiae. Second is beer. The production of beer is a multi-step process designed to break down the starches of grains such as barley to produce simple sugar, which can then serve as a substrate for alcoholic fermentation by yeast. Third is distilled spirits. Distilled spirits are produced using distillation to purify the alcohol generated during fermentation. Fourth is vinegar. Vinegar is a product of an oxidation of alcohol by the acetic acid bacteria, Acetobacter and Gluconobacter. And lastly is the bread. In bread making, the carbon oxide produced by yeast causes bread to rise and the alcohol is lost to evaporation. Next is changes due to mold growth. Molds contribute to the flavor and texture of some cheese. As already discussed, in addition, many traditional dishes and condiments used throughout the world are produced by encouraging the growth of molds on foods. Succession of naturally occurring microorganisms are often involved. The microbial and chemical aspects of many of these traditional foods have not been extensively studied. First is the soy sauce. Soy sauce is made by allowing species of Aspergillus to degrade a mixture of soybeans and wheat, which is then fermented into brine. So let's move on to the food spoilage. So food spoilage encompasses any undesirable changes in food. Spoilage microorganisms under microbial metabolites that cause repugnant tastes and odors, although aesthetically disagreeable, generally are not harmful. This is not surprising when their growth requirements are considered. Most human pathogens grow best at temperature near 37 degrees Celsius, whereas most food are usually stored at temperature well below the normal body temperature. Similarly, the nutrients available in fruits, vegetables, and other foods are generally not suitable for the optimum growth of human pathogens. As a result, the non-pathogens can easily outgrow the pathogens when competing for the same nutrients. Spoiled foods are considered unsafe to eat. However, because high number of spoilage organisms indicate that foodborne pathogens may be present as well. Common spoilage bacteria are the Pseudomonas, Erwinia, Acidobacter, alkalogenes, lactic acid bacteria, and endospore formers are important causes of food spoilage. A wide range of bacteria is important in food spoilage. Pseudomonas species can metabolize a wide variety of compounds and grow on and spoil many different kinds of food, including meats and vegetables. Psychrophilic species are notorious for spoiling refrigerated foods. Members of the genus Erwinia can produce enzymes that degrade pectin and so they commonly cause soft rot of fruits and vegetables. Acetobacter species can transform ethanol to acetic acid, the principal acid of vinegar. Although this property is very beneficial to commercial producers of vinegar, it presents a great problem to wine producers. Milk products are sometimes spoiled by members of genus Alkaligenes that form a calyx, causing strings of slime or riphonis in raw milk. The lactic acid bacteria, including species of Streptococcus, Leuconostoc, and Lactobacillus, are produce lactic acid. Anyone who has unexpectedly consumed sour milk knows that these can be disagreeable. Members of the genera Bacillus and Clostridium are particularly troublesome because their heat-resistant endospores survive cooking and in some cases canning. Bacillus coagulans and Bacillus erythromorphicus spoil some canned foods. So next is the common spoilage fungi. Fungi grow readily in acidic as well as low moisture environments. Therefore, fruits and breads are more likely to be spoiled by fungi than by bacteria. A wide variety of fungi, including species of Rhizophus, Alernaria, Penicillium, Aspergillus, and Botrytis, spoiled foods. Since fungi grow readily in acidic as well as low moisture environment, fruits and breads are more likely to be spoiled by fungi than by bacteria. Aspergillus flavus grows on peanuts and other grains, producing aflatoxin, a potent carcinogen monitored by the Food and Drug Administration. Now, let's move on to the foodborne illness. Commonly referred to as food poisoning, occurs when pathogen or a toxin it has produced is consumed in a food product. Consumers must employ sound preserving preparation and cooking techniques to avoid hazards from both home-prepared and purchased food products. In spite of the strict controls of the quality of the food supply, millions of cases of food poisoning are estimated to occur each year from food prepared either commercially at home or been prevented with proper storage, sanitation, and preparation. One challenge is to prevent foodborne illness is to develop reliable methods to detect pathogens before a contaminated product is marketed and consumed. Not only are cultured methods time-consuming, they often lack sensitivity because of the large number of competing organisms generally found on foods. Rapid methods utilizing antibody tests such as ELISA and DNA probes are being developed to detect many pathogens. Foodborne intoxication. Foodborne intoxication results from consuming a toxin produced by microorganisms growing in a food product. Foodborne is an illness that results from consumption of an exotoxin produced by microorganisms growing in a food product. When such a food product is ingested, it is a toxin that causes illness, not the living organisms. Staphylococcus aureus. Many strains of Staphylococcus aureus produce a toxin when ingested, cause nausea and vomiting. Although Staphylococcus aureus does not compete well with most spoilage organisms, it thrives in moist, rich foods in which the organisms have been killed or their growth has been inhibited. For example, Staphylococcus aureus can grow with little competition on unrefrigerated salty products such as ham. Creamy pastries and starchy salads stored at room temperature above also offer ideal conditions for the growth of this pathogen because the cooking of the ingredients kills most other competing organisms. The source of Staphylococcus aureus is usually a human carrier who has not followed adequate hygiene procedures such as hand washing before preparing the food. If the organism is inoculated into a food that can support its growth and the food is left at a room temperature for several hours, Staphylococcus aureus can grow and produce a toxin. Unlike the most exotoxin, Staphylococcus aureus toxin is heat stable so that cooking the food will not destroy it. So third is botulism. Botulism is caused by ingestion of neurotoxin produced by anaerobic spore farming gram-positive rod Clostridium botulinum. Unfortunately, a growth of organisms and production of a deadly toxin may not result in any noticeable changes in the taste or appearance of the food. Canning process for low acid spore is specifically designed to destroy the endospores of these organisms, but processing errors or post-processing contamination can allow the germination of the endospores and the growth of the vegetative cells that produce toxins. As an added safety measure, low-acid home canned food should be boiled for 10 to 15 minutes immediately before consuming to ensure destruction of potential botulinum toxin. Foodborne infection. Unlike foodborne intoxication, foodborne infection requires the consumption of living organisms. The symptoms of the illness, which usually do not appear for at least 1 day after eating the contaminated food, usually include diarrhea. Throughout cooking of food immediately before consumption will kill the organisms, thereby preventing infection. So the foodborne example is the Salmonella and Campylobacter. Salmonella and Campylobacter are two genera commonly associated with poultry products such as chicken, turkey, and eggs. Inadequate cooking of these products can result in foodborne infection. Cross-contamination of other foods can result in the transfer of pathogens to those foods. For example, if a cutting board on which a raw chicken was cut is then immediately used to cut up vegetables for salad, the salad can become contaminated with Salmonella or Campylobacter species. Next is Escherichia coli. 0157 oxygen 157 hydrogen 7. Escherichia coli causes bloody diarrhea that sometimes develops in hemolytic uremic syndrome, which is life-threatening. Escherichia coli has caused several large food poisoning outbreaks, including an epidemic in Japan in 1996 that sickened more than 9,000 people, a 1993 outbreak in the United States spread over 4 states. The latter epidemic was traced to undercooked contaminated hamburger patties served at a fast food restaurant. Raw meats are a troublesome source of foodborne infection, while the initial contamination bacteria such as Escherichia coli normally occurs on the surface and is easily destroyed by searing the exterior of the whole meat, such as steak. Grinding the meat distributes those same bacteria throughout the food. Prevention then involves more thorough cooking so that enough heat reaches the center to kill or the organisms. Next is food preservation. Preventing the food and concurrent metabolic activities of microorganisms that cause spoilage and foodborne illnesses preserves the quality of food. Some methods of food preservation, such as drying and salting, has been known throughout the ages, whereas other has been discovered or developed more recently. The major methods of preserving food: high temperature treatment, low temperature storage, addition of antimicrobial chemicals, and irradiation. So first, food preservation is canning. The canning process destroys all spoilage and pathogenic organisms capable of growth at normal storage temperature. Low-acid food are processed using streams, under pressure, or autoclaving in order to reach temperatures high enough to destroy the endospores of Clostridium botulinum. Acidic foods are not subjected to as stringent heating condition because Clostridium botulinum cannot grow and produce toxin in those foods. Second, food preservation is pasteurization, heating food under controlled condition at high temperatures for short periods of time, destroying non-swarming pathogens and reduce the numbers of spoilage organisms without significantly altering the flavor. Third is cooking. Cooking, like pasteurization, can destroy non-spore-forming organisms. Cooking obviously alters the characteristics of food. However, heat distribution may be uneven, resulting in the survival of organisms in adequate heated regions. Fourth is refrigeration. Refrigeration preserves food by slowing the growth rate of microbes. Many organisms, including most pathogens, are unable to multiply at low temperature. Fifth is freezing. Freezing stops microbial growth because water in the form of ice is unavailable for biological reaction. A portion of the organism will actually be killed by damage caused by ice crystals, but remaining organism can grow and spoil food once it is thawed. 6 is dry or reducing the water activity. Drying foods or adding high concentration of sugar or salts inhibit microbial growth by decreasing the ability moisture. Eventually, however, molds may grow. Seventh, lower the pH. Lowering the pH either by the addition of acids or by encouraging fermentation by lactic acid bacteria inhibits a wide range of spoilage, organisms, and pathogens. 8 is adding antimicrobial chemicals. Organic acids such as propionic acid, benzoic acid, and sorbic acid are naturally occurring antimicrobial chemicals that are added to a variety of foods to inhibit fungal growth. Nitrates are added to cured meat to inhibit growth of Clostridium botulinum and other organisms. Wine, fruit juices, and other products are preserved with the addition of sulfur dioxide. And lastly, the 9, irradiation. Gamma irradiation destroys microorganisms without significant altering the flavor of some food. However, its use is regulated. That's all. Thank you for listening.